going to talk about another aspect of glucose, and that's its role in post-translationally modifying proteins in a process known as protein glycosylation. And our interest in this work stems from our interest in inherited muscle diseases, particularly muscular dystrophies, and we identified a group of muscular dystrophies that result from inherited disorders of protein glycosylation, the addition of sugars to proteins. So I'm going to describe a bit of that work today. So just for a brief primer on muscular dystrophies, muscular dystrophy is an inherited disease most prominently affecting skeletal muscle, where the patients develop weakness at a young age and then typically get progressively worse over their lifespan, lose ambulation, and often have early death before the second decade of life. And the causal mutations that cause muscular dystrophy are complex. There's now more than 50 different clinical forms and at least 30 or probably 35 now genes that cause various forms of muscular dystrophy, and all of these genes are expressed prominently in skeletal muscle, and if they're in cardiac muscle, these genes will also cause highly prevalent cardiomyopathy. So for example, in the muscular dystrophies that we study, clinically detectable cardiomyopathy is detectable in about nearly all of the patients, and about 20 to 30% of the patients will actually die from cardiomyopathy, with the rest typically dying from respiratory failure or complications of respiratory failure. So I'm going to focus our attention on a subset of these muscular dystrophies that are associated with this complex here, the dystrophin glycoprotein complex. And this complex is a complex of glycoproteins that are present on the surface of muscle cells, including cardiac muscle cells. And these glycoproteins are named because they're attached to the protein dystrophin, which was the first muscular dystrophy gene described a number of years ago in Duchenne muscular dystrophy. The complex is composed of a number of glycoproteins. The one I'm going to focus on today primarily is the central protein in purple, dystroglycan. Dystroglycan is a 2-subunit protein with a transmembrane domain beta-dystroglycan, and then alpha-dystroglycan is an extracellular protein that binds to the extracellular matrix through a glycan-dependent interaction with laminin and other proteins present in the matrix. Now, dystroglycan has long been known to play a central role in this complex and act as the important extracellular matrix receptor, but quite surprisingly, we have identified very few mutations in dystroglycan. The first mutation was identified just a year ago, and confirmed that dystroglycan certainly is important in muscular dystrophy. But the reason that we got started in this a number of years ago is that mutations were identified in a number of genes that actually process dystroglycan during its secretion or movement to the plasma membrane. And these are all, we don't know what all of these genes do, but many of them appear to have homology to other proteins known as glycosyltransferases that transfer sugars to glycoproteins as they transit through the secretory pathway from the ER to the Golgi and ultimately to the membrane. And so this is sort of where we started a number of years ago in identifying the patients that had these forms of muscular dystrophy. And the key experiment is shown here on the left, where we've stained alpha-dystroglycan here in a normal patient. In skeletal muscle, the dystroglycan exists at the sarcolemma, and this antibody particularly was known to recognize a glycosylated form of alpha-dystroglycan, and this antibody staining was missing in these patients. Probably the most critical experiment was that we generated an antibody that recognizes the core alpha-dystroglycan protein, and when you run this protein on a gel, it's shifted in molecular weight by almost 50 kilodaltons. So about 70 kilodaltons of dystroglycan's molecular weight in muscle is glycan, so this represented a significant loss of glycan on the alpha-dystroglycan protein. And we had known at the time that the binding of dystroglycan to laminin was glycan dependent, and we went on to show that patients with these types of diseases have abnormal dystroglycan binding. And so, this disease has become increasingly complex because we've now, not our laboratory, but the field has generated has identified a number of mutations in a number of different genes that all appear to cause the exact same biochemical defect in alpha-dystroglycan that leads to its loss of function. We also know that there's using complementation studies in work done by Kevin Campbell's laboratory, that there's probably at least 5 or 6 other genes that all participate in this pathway. We don't know how they all work, and we don't know particularly how they all converge on this very similar important glycosylation event that leads to the functional activity of this protein. I will focus a little bit on this enzyme LARGE, which appears to be a key enzyme in this pathway. It adds a glycosaminoglycan-like chain to a dystroglycan that has a fairly long length and adds a significant molecular weight to the protein. It's a unique glycan, though, that's doesn't appear to be found on any other proteins other than alpha-dystroglycan. The other thing that we don't really completely understand yet is whether or not all of the disease phenotypes that are present in these types of patients are due to dystroglycan deficiency. We've made quite a bit of progress in this using tissue-specific knockout mice to knock out dystroglycan in specific tissues to try to determine whether dystroglycan is important for this phenotype. One of these experiments is shown here, where we took the dystroglycan floxed mice and crossed it with 2 different Cre lines, one to drive expression of Cre in ventricular tissue using the ventricular myosin light chain 2 promoter, and then also to knock dystroglycan gene out in smooth muscle, because there had been some suggestion that the DGC might be important in vascular smooth muscle as well, leading to a cardiomyopathic phenotype. And then we compared these mice to determine the effects of dystroglycan on the cardiovascular system. And what we showed is that knocking out dystroglycan in smooth muscle had no cardiac phenotype, but knocking out dystroglycan specifically in cardiac myocytes resulted in a fairly significant cardiomyopathy characterized by focal myocardial necrosis, as well as focal replacement fibrosis, shown here with serious red staining, with the contrast being fast green, labeling the myocytes. So this cardiomyopathic phenotype that's present in these knockout mice appears to be consistent with a functional dilated cardiomyopathy. This can be demonstrated by not only morphological studies, but also using functional studies. I'm also the director of the phenotyping core at the Cardiovascular Center, so this is the advantage of being in our cardiovascular centers, having access to these types of experiments. And so we've functionally characterized these mice, and these mice have a loss of ejection fraction that's consistent with a dilated failing heart phenotype. This, I should have pointed out on the previous slide, is this is a fairly slowly progressing cardiomyopathy in mice. Young mice don't have any detectable dysfunction or pathology. It appears approximately around 6 months of age, and it progresses to about 10 months of age. These mice do, the cardiac-specific knockout mice do die a little bit earlier than their wild-type litter mates, suggesting that this cardiomyopathy can be lethal, at least in some animals. So to study the mechanisms of what causes this cardiomyopathy in mice, we turn to younger animals prior to the onset of pathology. And one of the important phenotypes that's been observed in skeletal muscle is that muscles become susceptible to mechanical damage during skeletal muscle contraction. And to study this in cardiac muscle, what we did is we injected animals with a dye label called Evans blue dye, which normally binds to serum albumin, and so it doesn't normally cross membranes because it's bound to protein, but in cells that become damaged or leaky to Evans blue dye will accumulate in the cells, shown here in red. And what you can see is that the Evans blue dye accumulation in these dystroglycan knockout mice appears to be sensitive to whether or not these animals are exercising. So if you exercise the animals, you get more dye uptake in the heart, suggesting that there's a mechanical linkage to how this occurs. The other evidence for this mechanical linkage is that if you note the orientation of the Evans blue dye uptake in these patches of myocytes that get damaged in the myocardium, they appear to align along the mechanical axis of the cells and not spread into the neighboring cells that are in the perpendicular orientation to these myocytes. So this results suggested that cardiac muscle from dystroglycan deficient animals are sensitive to, potentially sensitive to mechanical damage that leads to disruption of the cells. And this may explain then why we have focal areas of myocardial fibrosis that progressively accumulate in the muscle, leading to its overall cardiomyopathic phenotype. And so in order to test whether or not this effect on cardiac muscle cells was due to a sensitivity to mechanical activity, we devised some new experiments to test acutely in isolated cardiac myocytes the effects of mechanical strain on cardiac myocyte stability. So this uses a tissue culture chamber that has a deformable surface on the bottom of the tissue culture plate, and we can deform this by lowering the pressure below the well and stretching the membrane. This stretches any cells that are attached to the surface and can basically mimic a beating heart in terms of its mechanical activity. This can be done in an incubator over hours or even days under the right conditions. And so what these studies, then we do the experiments, in the presence of a dye that doesn't normally get taken up into cells, but if cells become leaky, they will get into cells, and we use propydium iodide because it will bind to DNA and bind irreversibly inside the cell if the cell becomes damaged. So what you see under these experiments, if you stretch dystrophic adult cardiac myocytes, is that the cells start to take up the dye and the nuclei become fluorescent. You can quantify this in two ways. One is to look at the dye-positive cells that become dye-positive under stretched conditions. Many of these cells will also not only take up dye, but they undergo permanent cardiac contracture likely due to calcium influx leading to contracture of the cells. So both dye-positive cells as well as round cells increase, and this is significant in dystroglycan deficient animals. This model we used here is actually the MYD mouse, which is the mouse model of large deficiency, a true glycosylation deficient muscular dystrophy and cardiomyopathy. So we've gone on to show that this fundamental phenotype of sarcolemma instability appears to be a common mechanism in several models of muscular dystrophy with disruption of the dystrophin glycoprotein complex. So this is data using the MDX mouse model, which is a mouse model of dystrophin deficiency, which is the generally used model of Duchenne muscular dystrophy in humans. And what you can see here is a little bit different data in that in the MDX cells, the cells even prior to stretch are leaky to the dye uptake. But then when you stretch the cells, many of these cells undergo permanent contracture, likely due to calcium influx into the cell, leading to contraction of the cell. We've also now recently extended these studies to another model of dystroglycan dysfunction, and that is that there have been dominant mutations in DGC proteins, particularly one of the proteins of this sarcoglycan complex known as delta sarcoglycan. And when we express dilated cardiomyopathy mutant delta sarcoglycan proteins in cardiac muscle cells in a dominant fashion now in adult rat cardiac myocytes, we can again render these cells susceptible to mechanical damage. So in this case, in stretched cells, we see a reduction in rod-shaped cells, an increase in round cells, and all of these, nearly all of these cells that are round are also positive for dye uptake, indicating that they've undergone loss of sarcolemmal stability and calcium influx, leading to cardiac myocyte contracture. So we've spent a lot of time trying to develop assays to study cardiac myocytes in vitro, and of course one of our long-term goals is to use these approaches and physiological measurements to do high-throughput screening for small molecules. But when thinking about this, we have a couple of issues, what I call a mouse problem and a myocyte problem. So one of the mouse problems is that the dystroglycan knockout mice are embryonic lethal, and so this means that nearly all of the gene mutations that have been identified in each of these glycosyltransferases are also embryonic lethal. We are fortunate enough to have the large mutant mouse that has deficiency in the large gene. But the other problem is that on the other end of the scale, the MDX phenotype, which is the model of Duchenne muscular dystrophy, the cardiac phenotype in this mouse is extremely mild. So we have, in one case, gene mutations that are lethal. In another case, gene mutations are too mild. The other problem, as many of you know, with studying adult cardiac myocytes, is they're notoriously unstable in primary culture. So if you don't do anything to the cells and just put them in primary culture, the cells are all dead in 24 hours. We've done some cute tricks to try to make this a lot better. Unfortunately, this uses inhibitors that inhibit cardiac muscle cell contraction. So this prevents us as using the contracture as an important readout phenotype. And also we've found more recently that this inhibitor also inhibits an important membrane repair pathway in cardiac muscle cells. And so we're not too crazy about that model anymore of studying cardiac myocytes, at least for muscular dystrophy purposes. And so that's where I'll end and leave with a brief description of what we're proposing to do in the new study funded by the Technion-U of M collaboration, where we'd like to do similar to what was previously described in the previous talk about with inherited arrhythmic disorders, is to use embryonic stem cells to study now rare inherited diseases that are associated with or that cause muscular dystrophy. And so one of the unique features of this collaboration is it takes advantage of some very excellent teams at both institutions. And one unique thing that we have at Michigan is we have a human embryonic stem cell institute who has a collaboration with a company called Genesis Genetics. And this is the largest prenatal genetic diagnostic testing laboratory in the country, and actually does nearly all of the prenatal genetic diagnostic testing in the United States, as well as a number of countries. And so with this collaboration, we can actually predict how many human embryonic stem cell lines we can create from patient populations, and even for rare diseases such as Duchenne muscular dystrophy, for instance, which has about a 1 in 5,000 incidence. Genesis Genetics has screened more than 300 families in the last 3 years. So with our collaborators at Technion University, we hope to use these cells, both human embryonic stem cells and induced pluripotent stem cells, to derive muscle cells and neurons, and then to use the phenotypic screening that we've described in cardiac myocytes, along with our Center for Chemical Genomics, to do high-throughput screening for therapeutic molecules. So that wraps up my talk. I'd like to thank the individuals that contributed to this work. Most of the work that I described is by a talented postdoc, Yildiz Kabaeva, and we have a number of collaborators at Michigan, both in cardiac muscle and in skeletal muscle, and we look forward to our future collaborations with our colleagues at the Technion. And as you know, in Michigan, we have snow instead of sand, and so we strap things on our feet and go bombing down the hill for fun. So, all right, I'd be happy to take any questions.